Ladies and gentlemen, good afternoon or good morning, according to the place where you are living. I am Andrea Genazzani. Today is a pleasure to have with us Professor Gary Ventolini. Professor Gary Ventolini is an Italian born. He graduated in medicine in University of Padua. Then he moved and he joined the Liberal University in Cali, Colombia, and he was the chief of medical education. He had the postgraduate training in family medicine in Spartanburg in South Carolina, USA, and joined the faculty of the University of Cincinnati School of Medicine. Following an obstetrics and gynecology residency at Good Samaritan Hospital in Cincinnati, he joined the faculty at Wright State. He served as the chair of obstetrics and gynecology at Wright State University, Boone Shop School of Medicine, and is now the regional dean and professor of obstetrics and gynecology at the Texas Tech University Health Science Center at the Permian Basin. Is a very well recognized scientist, and he has devoted part of his studies in this last recent year on a very hot topic in the world now and in medicine. You know, we are living with the COVID-19 virus too, but we are also living with microbes. And with the microbes, we have made one kind of alliance together to survive in this world. And Professor Ventolini is now will make a presentation discussing its editorial and his paper, both published in the last issue of GREM, on current progress on vaginal microbiome, bacterial vaginosis, and biofilms.
It's an honor and a pleasure to be with you today. We're going to talk about the current progresses on vaginal microbiome, bacterial vaginosis, and biofilms. The discussion is going to basically be on regarding the vaginal microbiome. What's new? Regarding the vaginal immunology, we know that now that women are very well protected. Regarding vaginal bacterial vaginosis, why we have so many recurrences? And regarding the lactobacilli products, these, as you're going to see, are a gift for the future. I don't have any disclosure to do. I just work for the university and don't receive any other stipend anywhere else. So what is new on the vaginal microbiome? We know now that the vaginal microflora contains more than 200 species, and they respond to endogenous and exogenous factors, including the vaginal hygiene, habits, ethnicity, hormones, environmental, menstrual cycle, pregnancy, sexual intercourse, medications, and diet. We also know all that because of innovative molecular techniques that has permitted in-depth study of the microbiome, including the DNA fingerprinting, microarrays, qPCR, genomic sequencing, bacteria-specific sequencing of the 16S ribosome. And it is also important, sorry, I go back one, that the lactobacilli respond different to each antibiotic. We know now that there are some lactobacilli are very resistant to metronidazole, ciprofloxacin, gentamicin, clindamycin, and trimethoprim sulfamethoxazole. However, they are sensitive to the beta-lactamase, including erythromycin, tetracyclines, penicillin, and chloramphenicol. This is very important because when you prescribe any of these sensitive antibiotics to the patient for any reason, the female patient for any reason, you have to take in consideration that you may be altering their vaginal microbiome. The lactobacilli are the most abundant bacteria in women of a childbearing age, with a few exceptions all around the world. they derive from intestinal microbiome. So my question that I usually ask to the student is how the lactobacilli get inside the vagina. Basically, they are in the gut, and they have received some genes, acquired some genes through the through FOBs that has allowed them to use a system called vaginotropism, or a gene called vaginotropism gene, that they can migrate from the anal area and colonize the vagina. The lactobacilli are obligated on hormofermenters. Glucose from the vaginal epithelial cell is used to produce lactic acid and other products. The most commonly retrieved are Lactobacillus crispatus, and you can see here one of the crispatus actually in a wet mount with normal saline. The Lactobacillus gasseri is a little bit smaller. The Lactobacillus inner is shorter of all the these four lactobacilli, and Lactobacilli jenseni is a little bit thinner and shorter than the crispatus. It's very interesting that the crispatus, jenseni, and gasseri had the same genome. However, the Lactobacillus inner has a different genome, and that different genome has allowed them to be able to survive in a so-called healthy vagina and the one that is infected or has bacterial vaginosis. The healthy vaginas, therefore, contain mostly crispatus, jenseni, and gasseri. And you see them in culture and how do they grow. The lactobacilli present in the vagina, in the vaginal microbiome, go through daily modifications. Not everybody had just one bacteria, one type of lactobacilli the whole time. They can have a jenseni or gasseri or inners one day, and then the next day they have the majority of the other ones. However, the so-called healthy vagina, they have mostly the three that we have seen before, excluded the inners. The women are very well protected. Why? Because the lactobacilli make enormous amount of substances that are allowing to keep that vagina with 200 different species very, very quiet, we can say, just dominated by the lactobacilli. They maintain a balanced vaginal milieu between a pH between 3.8 and 4.4. They produce hydrogen peroxide, lactic acid, and bacteriocides. The bacteriocides are ribosomal antimicrobial produced. The lactic acid potentiate the bacteriocides and the hydrogen peroxide. And the hydrogen peroxide with the myeloperoxidase and chloride ions are potent toxic antioxidant, toxic oxidant bactericidal. The Lactobacillus crispatus and jenseni produce a fair amount of hydrogen peroxide. They are also antiviral. Some of the bacteriocides are protective against HIV infections. Vaginotropism, as we saw before, enabled by pili, so ligands for glycolipid receptors on the vaginal epithelial cells. They have a competition with the pathogen for the receptors on the vaginal epithelial cell. And they're also bactericidal through the lactic acid. They inhibit Neisseria gonorrhea. And through the hydrogen peroxide, they suppress the E. coli, the Gardnerella vaginalis, and the mobilumpus. And as you see, these are some of the current and just recent studies. The vaginal epithelial cells are very important because they provide exclusive microenvironment that helps to preserve the vagina and nurture endogenous lactobacilli while holding and acquire an innate immunity mediators. The immunity in the vagina is different from the general immunity. You can have a patient who is immunosuppressed, however, the immunity of the vagina is still working, or vice versa. The bacteriocins induce cell membrane permeabilization, ATR amino acid efflux, and transmembrane potential, and also the pH gradient depletion. So bacteriocins express host cell immunity, therefore they are no hemolytic or cytotoxic for anything else. And also the lactocin, 168, targets the cytomembrane of Gardnerella vaginalis. Here you see how the interaction of the lactobacilli are with the vaginal epithelial cell. They are very, very important to each other. Regarding bacterial vaginosis. Bacterial vaginosis is a polymicrobial overgrowth of gram-negative anaerobes and gram-positive bacteria that displace most of the lactobacilli, except, as we said before, except for Lactobacillus inert. How are they doing that displacement is still on the study. We have two studies going on that, and I hope that in our next meeting in November, being able to present you the way that the lactobacilli are basically displacing the Gardnerella and the Immobiluncus, especially the Gardnerella, to be able to colonize the vagina when you have originally some lactobacilli that are not the inners. It's important a little bit of historical synopsis, because we have, in our view, we have young physicians, middle age, and dinosaurs like myself. So in 1955, Gardner and Dukes identified the gram-negative, they were thinking bacillus in more than 90% of women with vaginitis, and they call it homogeneous vaginalis. And their vaginitis was basically the landmark of that was, and the sine qua non was to have the clue cells. Later, the Homophilus vaginalis was discovered to be gram-positive, so it was transferred to the Corynebacterium vaginalis. However, in 1977, Greenwood and Pickle show that the Corynebacterium vaginalis was both gram-intermediate and placed them in a new genus as the Gardnerella vaginalis. So today, when we talk about BV, bacterial vaginosis, we talk about the syndrome that bacterial communities, including some uncultivated species maybe that we haven't been able to find. Any of several of those 200 species of vaginal microbiome can cause these biopsies and bacterial vaginosis. And when the conditions are altered or reduced or blocked or deplete by the through the lactobacilli. So if you have the lactobacilli, if you are seeing a patient that has vaginal discharge and you put a wet mount in a normal saline on that very important instrument that is almost disappeared from everywhere, that is called microscope, and you see the lactobacilli present, then you know that the patient is well protected, and you have to find out what's going on. If they are lactobacilli, but they are very, very small lactobacilli and bacteria, then you know that here they have a bacterial vaginosis. If they have the clue cells, of course, then we have to come to the other criteria to diagnose anaerobic vaginitis, or and see, we'll see very, very well here that this is a bacterial vaginosis in which you have the clue cells, which are all squamous epithelial cells that have been totally covered by bacteria, and there are no lactobacillus, zero lactobacilli. And here we see, according to Gupta, how the healthy vaginal dominated bacteria the vaginal microbiome can go with the inners that are making the problem, or the ones who had learn how to live on normal and infected vaginas. And when the overgrowth is there, you have this biosis, and therefore the bacterial vaginosis. The lactobacillus inner is important also in other kinds of patients that had other medical problems, like when you have obesity and you have recurrent vaginal bacterial infections in women of reproductive age. We have demonstrated that in a recent publication in the postgraduate medicine, that obesity is associated with the lactobacillus inners and interleukin-1 alpha, interleukin-6, interleukin-12, and interleukin-17. So you'll see, beside all the possible problems that are related to the pre-existing conditions, we have to think that also obesity is doing a part of that. What are some of the enigmas that we have regarding bacterial vaginosis? Why there is no evidence of inflammation in bacterial vaginosis patients? And the second, why there are so many recurrences? Is a major overgrowth of the gram-positive and gram-negative bacteria and the pro-inflammatory cytokines, that, as you can see there. Is that immunotolerance from co-evolution? Maybe. Those bacteria has been on the vaginas until since the non-human primates were becoming humans. Is because of poor antibiotic penetration? Could be. Is because of alterations on the bacterial physiology? Or is there infection? Today, we know that is all of the above, and also the biofilms. The bacteria, through the evolution, they have learned to defend themselves by producing an extracellular matrix from which they can protect themselves, that is called biofilm. Here you see the representation of the biofilm with the lactobacilli inside. This is by courtesy of Dr. Hamoud, who is my co-author in the editorial. And here we see we were the first one to publish actually the biofilm formation by vaginal lactobacilli in vivo. This is very important because we send, in science, we have to have an open mind always. This paper was rejected many times. And then we send it to the medical hypothesis, and medical hypothesis published it after we demonstrated that in vivo. So sometimes perseverance is the best answer to medical dilemmas. We get in contact with the Institut Pasteur in Paris because we needed to have a way of quantify the amount of biofilm produced and be able to work with it. So the Pasteur Institute had a patent for these microfermenters, in which you'll see you grow on the left, you grow the biofilms, and then you are able to extract the spatula, and through the spatula you are able to see and retrieve the biofilm and be able to use it for the experiments. These instead are just the biofilm. If you put a Gardnerella vaginalis and you add a supernatant on Lactobacillus gasseri, you knock down completely, almost completely, the biofilm produced by Gardnerella. the reduction has been close to 90%. So then we asked the question, can they work also against other pathogens? And like anything in science, we have one of our researchers, Aitiya Hamad, that one day in the lab, she said, oh, what happened if I put this supernatant on Pseudomonas aeruginosa? And wow, look at what happened. So the supernatant using Lactobacillus gasseri completely knocked down the Lactobacillus, the Pseudomonas aeruginosa biofilms. And here you'll see also the ones that were retrieved from in vivo. So then we said, okay, if in vitro happens like that, what will happen on real mice? So we took some mice, and what we did with the mice was produce thermal injury and dorsal excision. And we took the supernatant and injected the Lactobacillus gasseri 63 AM supernatant to the mice, half the mice, and the other half the mice received just an injection of normal saline. And we see here that the Lactobacillus gasseri inhibited the Pseudomonas aeruginosa strain PA01 growth. It prevented the biofilm development and also eliminated partially it developed all the developed biofilms. On the thermal injury model, a single injection of Lactobacillus gasseri after injury and with the strain applied with the strain of Pseudomonas aeruginosa infection, reduced the mortality to 0% and prevented sepsis. The second injection after 24 hours eliminated completely the Pseudomonas from the wound. On the murine dorsal excision infection model, either the Lactobacillus gasseri of ceftazidime treatment with the infection by the Pseudomonas aeruginosa wound significantly reduced the mortality rate in mice, while combining both with ceftazidime eliminated completely the mortality. So I think that this study was also recently published in the Journal of Medical Microbiology. So you can see that we have some hopes for the future. So the current and future developments, the identification and isolation of that Lactobacillus gasseri 63 AM products that helps to displace or produce the that are responsible, excuse me, of this Pseudomonas aeruginosa biofilm obliteration. We are going to look for the FDA application for human usage in Lubbock, Texas, where our main campus is for Texas Tech University Health Sciences Center. We have a wound unit, a regional wound unit in Texas, and also then start a randomized clinical trial in patients who are severely wound infected, because as you know, the infection kills a lot of them. More than 150,000 persons died in the United States because of wound infections by or sepsis by Pseudomonas. They are super bugs, so we don't have a good bacteria, a good antibiotic to help them. They could be a combination of antibiotic plus the current treatment with the byproducts that we are identifying now. And also we are studying the displacement that the Lactobacilli are doing on the Gardnerella vaginalis, so we'll be able to better understand why some patients get infected and reinfected and keep getting infected. So I think that I was very fast. I talked very fast, but we updated the vaginal microbiome, what we know so far. We appraised why the women are very well protected. We discussed some of the bacterial vaginosis enigmas, and we recognize that we have a gift for the future given to us by evolution by the lactobacilli. Thank you very much for your attention.
Thank you. Thank you very much, Gary. And then now we go to the question and answer. We have received several questions. First of all, I would like to thank you for the beautiful presentation. We have a huge number of people with us. And then I would like to start with a very simple and clinically important question when we speak about the vagina. Is vaginal douching still recommended?
No. And why is no? Now we know that the vagina is a cavity that has 200 bacteria. So do you want to remove the good ones, the lactobacilli from there? No. It's very interesting because in Europe, we still see in the hotels that they have the bidets for having the cleaning. So if you want to clean yourself, then you have to use an acidic solution. And what is the best acidic solution? It's called acetic acid, therefore vinegar. And it has to be a white vinegar, not balsamic vinegar. How do you dilute it? 2 tablespoons, 12 ounces of water, and then you can have outside cleaning. If you want to clean inside, you can take a swab, put it inside, clean, and then you are supporting the vaginal microbiome.
Thank you for this nice question and suggestion, and easy suggestion. Then we have a question from Sharvari Ozalkar, and he's asking, what is the role of vaginal microbiota in pregnancy complications?
Yes, we have seen that the pregnancies who are complicated, for some reason, they have mostly lactobacillus inert. So if you have a dysbiosis in the process of trying to get pregnant, it's going to be very difficult. If the dysbiosis is produced after you got pregnant and you acquire that mostly those lactobacillus inerts, they are going to be more involved with your possibility of having an early pregnancy loss or having a miscarriage. And also, if you have it after, you will have more tendency to have postpartum infections. And we are going to publish that very soon. I was asked to look at all the studies that show that. So very soon we're going to have that publication that is going to explain at each one of the process of the pregnancy from preconception to postpartum, the importance of having a microbiome that is working.
Yes, and also we have also an anonymous who asks, what do you recommend for bacterial vaginosis recurrence?
The first one is that one. Try to acidify the vagina. You get the swabs. What we do is, first of all, we change the type of antibiotic that we're using. If you use metronidazole, and I hope don't have anything to do with the metronidazole, then you go to nizoral. Second, you're going to get the swab, and 2 times a day, you're going to get your solution, very diluted solution of acetic vinegar, and then clean yourself. And then make sure that your partner is also well aware of that. So certain partners, we don't have other problems, other partners are their hygiene is not the optimal one, and then you go from there. If you use too much soap, it's not good. So at the end, rinse with a little bit of very, very diluted acetic acid. That will help.
Thank you. Can you repeat it, please, because for our for the people who are with us, how you do the percentage of the dilution of?
Yeah, it'll be 2 tablespoons of white vinegar. The cheaper, the better, because it's more pure, and 12 ounces of water.
And 12 ounces of water. Fantastic. Yes. Okay. Then we have also one question now rising from Selma Abdellawi. She asked, what kind of food promote the vaginal dysbiose?
Yes, usually if you have a diet that is very rich on starch, that is going to be implicated there. If you have a food that has a lot of meat, so you have to have balanced food, basically, the balanced diet. A balanced diet yes. is always the best. So you have to have a little bit of everything, like we learned when we were in medical school. The most important thing is the soaps are very bad. The bubble baths and things like that, those are interfering with the vaginal growth of that microbiome. Another thing that I have to fight sometimes with the patients is shaving. The hairs were made there by the evolution for a reason. That's where the lactobacilli can migrate with the vaginotropins genes that they have and colonize the vagina. But of course, I don't want to fight with women regarding shaving or not shaving. So I told them, if you have too many infections, try this one, let it grow for a little bit and see if you are better like that. And then you will thank me or hate me.
Then we have another question. Why Fan Lam? She or he, I don't know, because my term to better understand the Oriental name is not very easy. She asked, how can we restore the lactobacillus in vagina in women with recurrent bacterial vaginosis?
Yes. After you have treated properly, then you know they're going to grow from the gut, and they are there in the gut. And if you have it in the gut and you're not taking antibiotics, then they're going to and they have the way of get the pathway from the vagina to the rectal area, anal area, yes. Acidified, they will migrate. Taking lactobacillus, you can take it, but they have to be alive. The thousands and millions and billions of dead probiotics are not going to do it. So the best probiotics has to be alive probiotics that have alive lactobacilli. And those ones you'll take it after you have finished your antibiotics. And then you have the reserve that you need. They will bypass the acidity that is in the stomach. You don't have to take too many of those ones. So the best way to do it is in that way. You have several different products that has alive lactobacilli. Yogurt is not good for that reason, that first you make the yogurt and then you pasteurize. But kefir, you make first the kefir and then you add the lactobacilli. So kefir has alive lactobacilli and is available everywhere. It's inexpensive, a couple of tablespoons a day. There are a lot of flavors, a lot of brands and things like that. So that's the best way of replenishing your body from lactobacilli.
Thank you, Gary. Now, 2 questions related to the candidiasis. The first one is from Nadia Mello. What's the role of vaginal candidiasis along with lactobacillus in the microbiome? and also during antibiotic treatment. And the other related question is, what is the best management in vaginal recurrent candidiasis for pregnant and non-pregnant women?
Okay, that's a little bit more problematic, because basically the lactobacilli, when you have their fungi present, they don't disturb each other. They don't. So the best way is to treat the candidiasis in the way that we usually treat it. Sometimes we have to have a frequent vaginal suppository or orally for a certain period of time, making sure that your immunitary defenses are in good shape. And then start taking the lactobacilli by mouth. Unfortunately, they are a different category of microbiomes, so they don't fight each other.
Then another question going on another related area. Does this biosis also caused by urinary tract infections?
Yes. Remember that, let's say you have a urinary tract infection and in the vagina, and the way that you clean yourself is just to try to interfere with the pathway between the anal area and the vagina. So when you are treating your urinary infection, you have to make sure that at the end of treatment of the infection, you have to take some kefir. The kefir is inexpensive. They add the lactobacilli after the process was done, and that's the way that you can reacquire your normal vaginal flora. You allowed the vaginal flora to be acquired through your digestive system. That is the best way, instead of putting anything inside. When we have tried to put lactobacilli directly into the vagina, they survive very little for some reason.
And then now still, that question is from Begonia Martinez de Tejada. She said, okay, we know that having good lactobacilli is good for pregnancy. And how can the women get them during pregnancy?
How do they can get what?
The good lactobacilli.
Oh, when you have pregnancy, the lactobacilli are a little bit challenged by the pH that comes out to be a little bit more basic than the acidic that you need. So what you should use is use, first of all, diluted vinegar, white vinegar, to clean yourself, 2 tablespoons in 12 ounces of water. And the second one is to get the lactobacilli orally by using the kefirs. That doesn't is indicated in any diet, doesn't have any contraindications in pregnancy or not, and making sure that you do it more frequently. But usually a couple of tablespoons of kefir. If you don't have any problem, once a week. If you have any condition that is predisposing you to have fungi on the vagina, then or bacterial vaginosis, then you take it every day, and in that way you are replenishing your intestines with the good lactobacilli. The other thing is resist the urgency to shave.
Yes, and then now we have, sorry.
Hairs were placed there by the nature for a reason. Don't fight the nature.
This we have to do. We have to follow nature. Then we have two questions which are one related. One is which antibiotic would you prefer in bacterial vaginosis? That one. And the other one is related, first that one, which antibiotic would you prefer in bacterial vaginosis, and how to decide what kind of antibiotic is the best?
Yes, when you have bacterial vaginosis, the problem is not what kind of antibiotic you use, is how do you prevent after the antibiotic is finished that it recurs. Because any of the antibiotics works pretty well. The metronidazole or nizoral, they work pretty well. However, what you have to do is, as soon as the antibiotic is finished, is when you start again taking your lactobacilli by mouth, because that is what is going to help to change your vagina. Get your clean yourself with the diluted acetic acid, diluted vinegar, and take the kefir, and make sure that your significant person that you're having sex with gets also some cleaning, whatever a man or a woman. And that will help. The immunity of the vagina, remember, is different from the general immunity. So
yes, and then we have, first of all, you can answer to Ivan Larenas. He asked tablespoon or teaspoon. You have already tell are 2-3 teaspoon, not a tablespoon.
Yes.
Okay. And then another question from a friend of us, Professor Cuauhtemoc Chelis Gonzalez from Mexico. He asked, how should I treat the vaginal burning if I do not have any positive culture from any germ?
Just vaginal burning with no...
apparently no positive culture. Then as a sensation, as a personal sensation of burning, apparently not dependent from any infection.
The vaginal burning, you have to make sure that that person is not using the underwear that has any scent or any perfume. So using cotton, making sure that when you are at home, you don't and you are able to do it, wear just a shirt with no let air going there, and and also making sure that you have culture for uroplasma, mycoplasma. Sometimes uroplasma, mycoplasma, they don't come in the same culture unless you ask for, and then you have to treat those ones, and after the treatment of uroplasma, mycoplasma, then that will disappear probably.
Yes.
So look for other causes of that burning. The burning can come also from anything, from atopic dermatitis to the scent that they are using, the soap that they are using. So they have to look at all the...
Thank you for your noting what uroplasma and mycoplasma are doing. This is also an answer to the Rejan Tattelboom question. And now I have a question. How do you see the future utilization of lactobacilli?
Oh, we are now trying to find exactly what in the supernatant that we are using to get rid of the Pseudomonas aeruginosa. We try to find out exactly which is the product that caused that. So the future is going to be to synthesize that or discover exactly which is that product that is a bacteriocide that is produced, is something else, and then we can use it as an antibiotic therapy, together with the antibiotic or by themselves. So the good thing about this one is that the chances to produce any side effect is going to be very little, because it's a natural product. It's come already from the vagina, from bacteria that has been in the vagina for millions of years. So the chances that they produce any kind of rejection is going to be very, very little. So the future that we're looking at that is that one, basically.
Okay. And then we have a question, we have a few questions. They ask, what about vaginal probiotics, oral probiotics or vaginal probiotics?
The best probiotics has to be oral and has to be alive, as we saw. The cheapest one, less expensive, is kefir. The other ones, you have to give billions and billions and billions. The question is, why do you have to give billions and billions and billions if they were working? In the vagina, the problem is they work for 2-3 days and that's it. The impact that they have on the pH of the vagina is directly proportional on the amount that you put there. So kefir is a product that you find all over the world. There are brands everywhere. And it's a product, like I say, they produce the kefir and then they add the lactobacilli instead of the yogurts that they put the lactobacilli, but then they pasteurize. So the best way of that is taking it orally, making sure that that pathway between the vagina and the anal area is acidified, and then you get better. 30 to 40% of the patients that I see, and I see just very complicated patients, they get better just with this simple way of hygiene.
And now is another question, who put attention, after menopause, the vaginal situation is changing. And then what about the bacterial vaginosis treatment in postmenopausal patients and in aging patients?
Yes, in those patients, one of the most important things to do is to keep the vagina acidified.
Acidified, this is very
yes. So if you keep the vagina acidified, and then the lactobacilli will grow there. Keep the vagina healthy in the way of, in the postmenopausal, the squamous epithelial cells are reduced, so you have to keep it acidified, moist, and estrogen. Any ART, you don't have any contraindications with that, that's the best way of giving, any estrogen that you want to give it, with or without progesterone, depending on your situation. Topically could be, or just acidify it.
Yes, thank you, Gary. We have also some questions from Marilia and one from Noelia Ortiz Maffei, who ask about the use of boric acid. What is doing boric acid is good or bad for the biofilm, and this boric acid can be used in recurrent candidiasis?
The problem with the boric acid is very caustic. And when you have a caustic acid, you can make big burns on the vagina. So I usually keep the simple one, water and vinegar. And I usually tell the patients when they come to me, I'm going to give you this water and vinegar. It's 2 tablespoons in 12 ounces of water until you are 120 years old. And then when somebody calls you from somewhere and you have to go, you tell them, wait a minute, I still have to clean myself, and then you can go.
Yes. Then it's another question is related to the biofilm and to the biofilm, clinical use of biofilm disruptors in recurrent bacterial vaginosis by Marisol Fernandez. And another one was on the impact, how lactobacilli can affect biofilm from pathogens in the vagina.
Yes, what we see is the lactobacilli themselves are not very potent producers of biofilm, because basically when they have the acidity produced through the lactic acid and other organic acid and biosurfactants, they don't have to produce too much biofilm to keep the vagina healthy. However, when you have pathogens that are very highly producers of biofilm, then what you have to get is get rid of those pathogens, and then they will be able to take over again on the as the ones in charge of the vaginal microbiome again. So when you have a patient that is continually coming with a bacterial vaginosis, then you have to sit down with her and tell her, look, this is what we're going to do now. I'm going to give you treatment, but after the treatment, in order to restore, the most important part of the treatment for me on bacterial vaginosis is to restore the vaginal flora. And yes, the way that you restore the vaginal flora is by taking the kefir or any other product that is add the alive lactobacilli, because they are able to bypass the acidity also of the stomach, and in that way, and keeping your external genitals clean, and that's the way that you are going to do it. Remember that the certain women, for some reason, when they feel, they have the bad smell, they have the vaginal discharge and things like that, they try to use more soaps, different soaps, bubble baths, anything that they would like to get rid of their vagina, basically. Calm down. We have ways to gently taking care of your genitals and your vagina and replenish your vagina in the natural way, allowing your lactobacilli that have evolved for millions of years to take and do their job. Remember, if these four, actually three of these species, a part of the species, Gasseri, Crispatus, and Genazzani, are able to keep quiet 200 other bacteria for how many millions of women we have now in the world? They are doing their job.
Yes, and then now we have an interesting question from Hans-Peter Steiner. Did you know we have a series of other situations where the vagina is manipulated during medical acts? And he asked, do you recommend prophylactic local medication prior to embryo transfer? I would like to change that question. Then in another one, then did you believe that prior to an embryo transfer, we have to check what it is the population of lactobacilli present in the vagina, and then to decide to give some indication before to do the act of the transferring?
Yes, yes. That the second part of your question is the right answer. Before transferring embryo, before doing an intrauterine insemination, even check the lactobacilli. If you have lactobacilli, you can see. You just need a very expensive instrument called microscope. Take a drop of normal saline and look. And if you see the lactobacilli, you don't have to identify which one they are. If you have a variety of lactobacilli there, you are safe. You can go ahead and then do your transfer or do your intrauterine insemination.
This is a very important suggestion. And then another one is looking another phase of the diseases in women and is touching, what about women with diabetes? Are there certain things we should advise them?
Yes. When you are diabetic, then mostly the problem with the diabetes is your immunity, your general immunity is down, but when you have higher levels of sugar also in your vagina, then you are going to have a tendency to alter your vaginal microbiome. So, of course, the best thing to do is keep your blood sugar in good range. And second, make sure that you check your acidity. When you check your acidity on your microbiome, then you know on your vagina, you know that you have the good lactobacilli. The problem comes if you are trying to get pregnant and then you realize that you have your sugars are too high. Don't get pregnant until you, if it's possible, of course, 50% of all the pregnancies are unintended. That if your sugar is not under control, you're not going to have your vaginal microbiome prepared to receive the embryo in your uterus. And then you have more chances to have, of course, a miscarriage. The microbiome in the vagina is equal to the microbiome in the uterus. Now, with the studies that have been coming from Spain, yes, we are seeing there that we have a good microbiome in the vagina. We are going to have a good microbiome in the uterus also.
Yes, and then also now we have another question from Marilia. How to control the overgrowth of lactobacilli and how to control cytolytic vaginitis?
Okay. When you have lactobacillosis, yes, first of all, if you have lactobacillosis, you have to get rid of all the lactobacilli. So what do? You use any antibiotic that is susceptible to kill all the lactobacilli. Usually we use penicillin with sulbactam. So you give 7 to 10 days. That gets rid of all the lactobacilli. During that time, remember that whenever you use antibiotics, you have to cover for fungi. So you use any of the antifungals. I use fluconazole, 200 milligrams, 1 or 2 tablets. And then you get rid of those ones. And right away after it's finished is when you start giving the kefir, acidify the urine, the acidify the vagina, basically the genitals, and then you can proceed.
Yes, and then I would like, I thank you very much. We have a lot of approval from our public, and I would like to ask you a very general last question. We have a lot of colleagues looking at us, and then what Gary Ventolini is thinking about the vaginal hygiene in the young population, in the young fertile woman, and in postmenopausal time. How you would like that to suggest that we have to suggest to our patient for the vaginal hygiene?
Yes. In general, for the vaginal hygiene, you will say close your legs, and then you can use soap in front and on the back. Don't use foam in the middle. After you become, when you are pre-menarche, you are going to have problems mostly with the fungi, not too much with the bacteria. If you have problem with the bacteria, you have to think that there could be a external or foreign objects or something like that. The girls are very, very curious, and they put something on the vagina, and then you can have an infection based on that. On your childbearing age patients, the best way to do it is to use acidic solution. That's the way that you keep. You want to clean the vagina inside because there are some women that are obsessive compulsive. Take the swab, get the water and vinegar, clean inside, make sure that the cotton comes out, and then you are okay. Don't use any sprays or any, if you want to use any scent thing, do it above the suprapubic area. And for the postmenopausal, is the same. Try to keep that area clean using the water and vinegar. I think that when I started doing looking at the vagina almost 40 years ago, I should have patented the water and vinegar.
Okay.
And then we will be millionaires. It's a very nice product. It helps everybody. It's inexpensive. You can get it anywhere. the cheapest, as I'm saying, the better. And you don't have any bad odor from the vinegar when it's diluted so well.
Then repeat, please. 3 teaspoons.
2 tablespoons. 2 tablespoons in 12 ounces of water. I don't remember now the ounces converted to the
12 ounces. We will. In Italy, we have, in Europe, we have ml.
I should have made the conversion.
Okay.
12
Please.
ounces.
We will do. Each one of us, we will take we will play, and then
a jar,
yeah.
Big jar. And when you do that solution, it will last for a long time. You can use it in a washcloth. You can use it with the spray. You can use it just the squirt bottle.
Fantastic. And then
sorry for the ones who are just dealing with the recurrent infections, because then they're not going to have any more patients when they know that secret.
Okay. Then a last point, please. And during the rest, when the women sleep, they have to be covered, or they have to be dressed and covering with sleep their genitalia, or they can have night dresses but not using the sleep, because
yeah, they can. If they are able to don cover, it's okay. They can use cotton underwear. Try to don't use any cotton underwear that has any scent. Pure cotton underwear. And I'm sorry for the Gillette, but I'm against the shaving there. But I never fight with that. If you don't have an infection, you can do whatever you want. If you have an infection, then you have to be more careful. We have to help the lactobacilli vaginotropians to do their job.
Okay. Thank you, Gary. Thank you. I think that we have to thank Professor Gary Ventolini for that fantastic, highly scientific lecture expressed in a very simple way to touch the heart and the mind of our participants and to bring a strong message. You will find this beautiful editorial and this paper on Gram. And now I would like to remind to all of you, then the next month, the 19th of October, we will have another paper presented, and it will be another completely different topic. It will be an expert opinion on integrative treatment with inositol and lipoic acid for insulin resistance of polycystic ovary syndrome, and will be done by Professor Alessandro Genazzani. Then now, happy life, happy stay, healthy life in this COVID-19 world. And I would like to thank again Professor Gary Ventolini and all of you for being with us this 21 of September 2020. Goodbye to everybody. Gary, thank you very much. We will see in Florence, we will see by video. You know, yes. The ISG Congress has been completely moved to an online event. It will be a fantastic event. Thank you.
Thank you very much. Thanks for everything. Be safe.